So I want to introduce the first speaker, Preston. He's a serial and parallel entrepreneur in biotech and blockchain. He received his bachelor's in neurobiology from Cornell and did his PhD in genetics at Harvard from the lab of George Church. He has launched, managed, and advised multiple companies and nonprofits in the space, including the Personal Genome Project, Veritas Genetics, Nebula Genomics, Samplify Bio, Doc.ai, Telome, Mind First Foundation, and more. So it is my utmost pleasure to invite Preston to please come and share his work with us.
I'm here to talk about the evolution of genomics and the sort of technological advancements that are taking us in the direction of the use of what people call big data. So I'm, as Sunandini said, I'm the Chief Scientific Officer of Veritas Genetics. I'm also a director of the Personal Genome Project at Harvard Medical School, founded 13 years ago by George Church, my PhD mentor. And the Personal Genome Project is really sort of the incubator, the foundation for a lot of what I'm going to talk about today. So yesterday at Harvard Medical School, there was a seminar, MD versus Machine, How AI Will Transform Medicine. I don't know if any of you saw that, but I highly recommend it. It's on YouTube. And these are some of the claims that were made in the introductory part. And I agree with these. AI is fueled by availability of big data, advanced machine learning algorithms, growth in computational power. But as the talk went on, you saw that there were some other really critical developments that fueled AI. And these are some of the more in the weeds kind of determinants or underpinnings of AI, the things that need to be done, that need to be there for AI to work. There have to be clear goals, algorithmic goals. You have to control for bias. Bias is a really, really big problem that's inherent in almost every data and every goal that we're looking at. So in the data quality, we have to learn how to filter the data to avoid bias and avoid garbage in, garbage out. This is a huge problem in machine learning and AI. So in creating high quality data, we need references and standards. I know that's really kind of mundane stuff, but it's so key to the development of machine learning. And then across many different kinds of data sets, we need to normalize, harmonize the data so that we're not finding signals in the data that are actually anomalies between data sets. So I'm going to touch on some of the practical things that we've done to make advances in these areas. So the Personal Genome Project, as I mentioned, started in 2005. It has a very broad and open consent. This is one of the really important things about the Personal Genome Project that differentiates it from any other biomedical study in the world today. You see a lot of information, a lot of media coverage about really large biobank projects, databases that have hundreds of thousands of participants, but none of those can do a variety of things that the PGP can do. For example, they can't recontact participants. They can't get participants samples of various kinds. They can't directly interact with participants to get, say, a saliva, blood, or even a cell sample. So the PGP is really unique in that way. So if you're thinking about running a research study, the kind of consent that is provided by the Personal Genome Project allows you as a researcher to do all kinds of stuff you can't do with other data sets. So now there are, in addition to the Harvard PGP, there are multiple international PGPs, Canada, Austria, UK, China, others coming online with over 15,000 international participants. And the Harvard PGP has thousands of genetic data sets, surveys, phenotypes, and generates only whole genome sequence data. And I'll tell you why that's really important for the development of machine learning and AI. So Going back to the standards point, we, in the early days of genomics, realized the importance of standards for calibration or harmonization of data across many different generators of data or repositories of data. Clinical labs need to be able to show that when they turn on a machine or they turn on multiple machines and workflows, that they're producing the same kinds of data from the same samples. So the PGP has worked together with the National Institutes of Standards and Technology, NIST, to create what are called genome in a bottle standards. These are cell lines. They're generated from PGP trios, Ashkenazi and Chinese families, that clinical labs use to sequence the genomes, get the DNA, and then cross-compare to the NIST-generated standards, the reference, to show that they can generate exactly the same data from the same sources. Out of the PGP has come the development of the Common Workflow Language by the company CureVerse, which is responsible for the computing infrastructure of the Personal Genome Project. It's a spec for allowing efficient creation and linking of these genome scale workflows. And then I'll describe in a little bit tiling, which we're creating as hopefully a standard for the representation of compressed genomic data, computable but compressed genomic data. So I've been using this word genome. So a lot of people don't really know what a genome is. So this is a genome. It's all the genes and functional elements, about 20,000 protein coding genes, about over 20,000 non-protein coding genes, a few million regulatory elements. It's all DNA in an individual cell of your body. That's what a genome is. A lot of people who work in genomics and only know a genome through the computer will be surprised to find that when you sequence someone's whole genome, a genome is actually 6.4 billion bases. You hear and you read all the time in the media that a genome is 3.2 billion bases, but when you actually want to sequence a person's genome, you're dealing with that amount of genetic material because that's 23 pairs of chromosomes. You get one pair from your mother, one pair from your father, and so the total amount that we refer to, we typically use in discussions, and we have to because we use it in calculations, we use it in storage, we have to know how much information we've got is 6.4 billion letters in an individual person's genome. So you've probably seen this graph many times. This is a really important graph to illustrate multiple points. One is that that genome sequencing has been declining very rapidly. It's actually an aggregation of data from two different technologies. The top of the graph, very shallow decline, was sequencing done with old technology. The much more rapidly declining price in genome sequencing was due to what we call next generation or current generation sequencing. That now is dominated by a company called Illumina. You can see that the price, as it approached $1,000, has started to plateau. Veritas Genetics, one of the reasons that I was interested in founding Veritas Genetics is to try to break through this $1,000 barrier and below and increase the availability and the competition for low-cost genome sequencing. The plateau there around $1,000 is really an arbitrary decision by one company because they have established largely a monopoly in this space. So they've set the price, and there's little competition to force that price lower. Hopefully that's changing in the near future. This is a projection of the trend into 2019 and a little bit beyond that I think will occur based on that growing competition in the space. I think I'm very hopeful, but we'll see. So why are people interested in whole genome? Well, clinical geneticists are interested because relative to, say, only the protein coding regions, you can get more comprehensive diagnoses. You can actually, this is from April, you can actually get better than 50% diagnosis of certain rare diseases compared to kind of a standing diagnosis of 25% with exome. So we're actually doing more than twice as well as exome if you do whole genome sequencing. However, it's even though you're doing twice as well in clinical diagnosis, the amount of DNA you have to look at is a lot more. Again, individual genome sequence is about 6.4 gigabases, and exome about 100 megabases. It's about 1.6% of whole genome sequence. Gene panels, which are typically used for specific indications like cancer risk, are much smaller. The smaller number of genes, dozens or a few hundred for the largest panels. And then genotyping, which is the kind of test that is done by 23andMe or Ancestry.com, represents about 0.01% of the whole genome sequence, which is, it's 640,000 SNPs with single nucleotide polymorphisms on a genotyping array. So it's a tiny fraction of the genome. So as we move toward the use of genomes, because of this improved clinical diagnostic potential and for other reasons, we start getting into really big data. The amount of data that comes out of, so just to sort of cut to the chase, we're talking about 150-ish gigabytes of information in a whole genome sequence, and 4 million to 5 million variants per individual relative to the reference genome. And this is typically represented in some standard notation, standard file formats, where there's actually can be some ambiguity, which is a problem with going back to GIGO and the need for really clean and clear data. And when you're doing machine learning, you need unambiguous representation